Good morning, everybody. My name's John Cumbers, and I'm the CEO of SynBioBeta. I just got stuck in traffic, so that's why I'm having to take big pauses in between my sentences to pretend that I was more organized coming here this morning than I actually was, so excuse me. I'm going to pass the microphone along, and while I get my breath back, let the panelists introduce themselves. Didier, starting with you. Actually, you have mics already, so you're good.
Hi, good morning, everyone, and thanks for having us this morning. I'm Didier Toubia, co-founder and CEO of Aleph Farms. At Aleph Farms, we grow a whole muscle meat from cells we isolate from a cow. The focus of the company is on reproducing the same experience. Should I start from the beginning?
Yes.
Great, so nice to be with you today. At Aleph Farms, we grow a whole muscle meat. The idea is to be able to reproduce the same experience, texture, taste, and flavor of a whole steak, not just hamburgers. The company has started in collaboration with the Technion, which is an academic institution in the north of Israel, with Professor Levenberg, a leading researcher in the field of tissue engineering, and with the Strauss Group, which is a food company.
Hello, I'm Avi Rohit. I think this is working now. I'm a professor at UCLA, and I'm a biophysicist by training. I run a research lab that focuses on cellular mechanobiology. So we're very interested in understanding the physical and mechanical aspects that impact cell biology and processes like cancer and neuromuscular diseases. How did I get into food? Well, I've been passionate about food my whole life, and cooking, considered going to chef school, and realizing the parallels between my research and food, I started a course using food to communicate science to the general public and also undergraduate students, and founded the Science and Food Organization at UCLA about 8 years ago. And I've also been involved in advising companies like Beyond Meat on their advisory board. Recently, my lab got into using our knowledge of mechanobiology to develop scaffolds to support co-culture of fat and muscle cells towards the goal of cultured marbled meat.
So my name is Leo. I'm a professor at the University of Nebraska, Lincoln. I have a lab work on large-scale cell and tissue manufacture. So in the past 5 years, we realized that it's extremely challenging if you want to make, for instance, about 1 trillion cells. And people are trying to using like the stir tank bioreactor, microcarriers type of stuff, which were originally designed for the chill cell industry or the bacteria industry. So we think that the efficiency is not good enough. So we started to develop our own technology, which is, I don't have slides to show them, so it's a very interesting, like an edible microtubule bioreactor, and then we can increase the cell culture density to about like 500 million cells per mil of volume, which is close to the tissue density, and that allows you to make a large number of cells within a small, tiny volume. The second area we try to address is if you have a lot of cells and you want to make a large volume of tissue, how you do that? You need scaffold, and more importantly, you need the circulation system. So we developed this called edible circulation system. You can put them into the scaffold, or you can use themselves as scaffold to make a large volume of tissue. And then after the tissue, you just eat them. So it's edible and dissolvable. So that's my research.
Great, and Leo, we're actually going to show a couple of your slides in a second, so you can demonstrate what you're talking about. Liz.
Hi, everyone. It's a pleasure to be here. My name's Liz Specht. I'm the Associate Director of Science and Technology at the Good Food Institute. My academic life prior to joining GFI was kind of a mix between chemical engineering and molecular biology. And so food is really a new application area of some of these real fundamentals that apply across disciplines. My role within our science and technology team is really spearheading kind of the analysis section of our work. So kind of landscaping what the predominant technical challenges are within the alternative protein field, going and finding people who have the right academic background from these various disciplines to kind of fill the knowledge gaps in this space, and kind of articulating high-value research projects that can address some of the shared challenges being experienced by all of the cell-based meat companies, to really move the field forward on some of these pre-competitive research areas.
Excellent. Thanks to all the panelists. I didn't get a chance to say what SynBioBeta is, but SynBioBeta, for those of you who don't know, is a synthetic biology innovation network. And we run an annual conference here in San Francisco once a year. We send out a weekly newsletter, and we keep track of the whole of the synthetic biology industry, who's doing what, everything from reading, writing, and editing DNA up through the tools chain, the platform layer, and then right at the top we have the applications companies. And of course, the burgeoning field of cell-based meat is one of those application companies, is one of those application areas. Before I started SynBioBeta, and I started SynBioBeta in 2012, in 2011 I ran something called the Synthetic Biology Launchpad. And one of the first companies that we funded was Modern Meadow. So I'm still a shareholder in Modern Meadow. And for those of you who know the history of Modern Meadow, they started out looking at cell-based meats. Now they've pivoted into leather. But I remember at the time when the press release went out, my mum is on my mailing list. I'm sure maybe some of you have your mum following you and in your career as well. And I got a one-sentence reply from my mum when the announcement about Modern Meadow went out. And the one sentence was, well, one word, sorry, it was yuck, exclamation mark. But it is wonderful to be here 8 years on and to see really this very exciting and burgeoning industry. But the panel is going to be all about the technical hurdles which still remain for this to become a commercial success. So we're going to come on to some questions, and then we're also going to throw it out to audience Q&A. But before we do that, Didier, you have a couple of minutes of slides, and then Leo, you have a couple of minutes of slides. So we're going to just help you to set the stage. So we're going to start with, if we can bring up Didier's slides. And do we have a controller, or do we not need a controller?
We have a short movie.
We have a short movie, no slides.
Exactly.
Okay, great. So we have a short movie of Didier that I hope we can queue up and play when you're ready.
Once upon a time, the world was in balance. People used to grow their own food. They even called animals by their names. There was a connection between what we grew and what we ate. But today, in our hyper-industrialized world, we've lost this connection. We send billions of animals to slaughter every year. Using 30% of available lands, consuming 15,000 liters of water for every kilo of beef produced, increasing issues of contamination, antibiotic resistance, and climate change, throwing our world out of balance. But what if we could go back to growing food that was in balance with nature? What if we could grow a steak identical to the steak we all like, but without the downsides to health and the environment, and in a way that's more humane? At Aleph Farms, we believe meat should be celebrated and enjoyed. That's why we directly grow safe, cell-based real meat similar to farm meat, the same meat with an improved production process. Aleph Farms replicates the muscle regeneration process that naturally occurs inside the cow, but on the outside. Using the same types of cells found in a steak, we grow slaughter-free meat in 3D, ensuring an end product which obtains the same taste, texture, and structure, advancing towards our vision of creating a cell-based steak. We're entering a new era by growing meat that's good for you, good for animals, and good for the planet, leaving a better legacy to future generations. This is no fairy tale. This is a true story, brought to you by Aleph Farms.
Fantastic. Thank you, Didier. And this actually reminded me of the reason that I invested in Modern Meadow in 2011. It wasn't about the application. I think actually my first reaction to cell-based meats was actually yuck as well when I first came across it. But Andras Forgax, who's the CEO of Modern Meadow, he was previously at McKinsey, when he just laid out the sustainability and economic impact that this could have on the world, that's what really hooked me, and I thought that's something that I wanted to invest in. Leo, I think we have a couple of slides from Leo, so if we could bring up Leo's slides. And Leo, in particular, you were talking about the quality of the cells, which is one of the first topics that I want to come on to. But
yeah, sure. So this is my lab name. We try to address the scale-up problem and the large volume tissue fabrication. Can you go to the next slide, please? Yeah. So here's some numbers that in the biomedical field, like the human cell culture, the solid tissue need about 500 million to 1 billion cells per gram of tissue. So if you want to make a 100 kilogram meat, you need about like 10, 14 cells. And the current cell culture technology is for the pluripotent stem cells. So I'm talking about the human cells, but animal cells should be very similar to the human cells. The cell density we can reach is very low. It's roughly about 2 million or 1 million cells per mil. So it means you need a huge bioreactor in order to generate enough cells, 1 kilogram of meat. And then, by the way, the human cells, you cannot catch them in a large bioreactor because the bigger the reactor, the more cells will die. So it's very challenging if you want to make like 10, 14 cells. Say, like you need a bioprocess like this. Start with 100 million cells. You do passage 1, passage 2, scale up, right? So definitely every passage you need to kill about 20 to 30% of the cells. And the final culture volume is like 100 K liters in order to make that many cells. It's economically and technologically not viable. So if you go to the next slide, please, thanks. So what we try to resolve these problems, we develop a new technology called dissolvable, edible, and scalable 3-dimensional tubular microbioreactor. So instead of go big, we go small. This basically some hydrogel tubes about your hair size. It's hollow tubes made with arginine hydrogel so you can eat them. So you cut the cell in these tubes. Now the tube will create a microenvironment that the cell really like. So what is the consequence? The consequence, you can catch about 500 million cells per mil of these hydrogel tubes. Like if you look at the right up corner, that is the human cells. It is pretty much like a noodle soup. Right? If you cook them, you can eat them, but remember this is the human cells, okay? So yeah, if you do the calculation, now look at the right low table. You want to make 10, 14 cells, start with the same cell. Now you can cut the time from 60 to 30 days, the passage from 11 passage operation to 1 passage operation, and total culture volume from like 100 K liter to about 320 liter. So you can imagine that it can significantly cut the cost. So that is a technology. So if you go to the next slide, please, thanks. This is a technology we developed, try to make a large volume tissue. Now, the most important thing is you need a circulation system, you have a large volume. So we're using the same hydrogel tubes. We cut the cell between the tubes. Sometimes you add some scaffold or not. So if you look at the right corner pictures, you see how the dye perfuse through this large volume of hydrogels. And if you look at the red and the green stuff, so if you have the perfusion system, what the cell alive, the green cells, and the red cell is the one without the hydrogel tubes, means the cell dead. So it's just a proof of concept technology, okay? And the third stuff is we also make this recombinant fibrin matrix for tissue engineering. So we think fibrin is the best matrix for tissue engineering. So.
Great, thank you, Leo. So we're going to talk about quantity, quality, and cost. So Leo, from everything that you just presented, if there was one bottleneck to scale, to getting the quantity increased, what would it be for you?
Quantity, right? Quantity.
Quantity.
Yeah, I think it's the culture volume. So we cannot culture the human cell or animal cell in large volume.
Great. Amy, same question to you. If there were one issue around how to increase the yield, how to increase the quantity, what would it be for you?
Well, we're particularly focused on the scaffolds, which I think is a major challenge in the field, and developing ways to be able to scale those scaffolds to be able to support the growth of multiple cell types.
And Didier?
Yeah, I agree. I think the relatively robust processes to grow very large quantities of cells in a large steel tanks. On the other hand, there is no yet large-scale tissue bioreactor available in the market, and no processes which have been developed for large-scale tissues. At Aleph, we're really focusing on growing a tissue, so that's for us a significant challenge we're addressing, and we're actually designing and developing our own bioreactor to fill in that gap. So we're kind of inventing a new field in that space of bioprocess development for large-scale tissue bioreactor, and that's exciting. I think it's a wide space which I believe has a huge potential moving forward in many different applications, and we expect that what we do today for food will eventually come back to medicine a few years later, and also spill over additional applications.
Liz, you get to rise above and look down and see all of the whole industry. Are you seeing that the three answers that we got, does that capture what you're seeing for the rest of the industry, do you think?
I think they do, in that they're different answers, right? So I think the key cost drivers here, the key bottlenecks for scaling, really inherently exhibit this interdependency between all different aspects of this process. And I think that's what's really exciting about where the field is at right now, is that nothing's set in stone yet. If you talk to different cell-based meat companies, there's relatively little consensus around even some of the kind of core questions about the process, which rather than being sort of overwhelming or confusing, I think points to an opportunity that the whole landscape is open. And so we can take a really systems level view of, if you optimize one aspect of the process, how does that impact, your sort of design requirements for other stages of the process. One of the projects that we're coordinating right now for value for the whole industry is a sort of combined life cycle assessment plus techno-economic analysis to build a working model so that you can try to find what some of those relationships are. If you increase your cell density by a certain amount, how does that change your ultimate culture volume? Or if you increase the rate at which you can recycle media, how does that affect your upstream costs? And sometimes those relationships are not intuitive. Sometimes there's very surprising kind of nuggets of insight that emerge from projects like that. But these kind of working models then allow us to pull people from different sectors with this really deep expertise in biomaterials or in stem cell biology, and have folks kind of align on a shared set of target metrics across the process as a whole.
I think it's a very good point. If I may add and build on what Liz just said, I believe that the right way to approach the scale and cost issues is really to look at the whole system. Many times we hear about the need for developing a low-cost serum-free growth medium, which is definitely one of the key drivers for the cost of the end product. But I don't think that we can isolate the growth medium from the bioreactor, from the whole process, from the whole system we're building. And the approach should be system-based and not focused on one specific unit of the whole system.
And I think there's different solutions for different problems as well. Of thinking about the texture of fish is very different than steak, and so the combination and interdependency, like Liz mentioned, of the bioreactor design with the scaffold design, the media, the cell types, might be very system dependent.
I'm shuddering at all the variables. I'm glad I'm not in the lab with you, but I'm glad that you are.
But like Liz said, exciting opportunity, right? Yeah.
Are there any hardware providers out there who are just focusing on this new market of cell-based ag?
Not exclusively, as far as I'm aware. So what's exciting is that there are multiple other industries that we can kind of look at their systems, how they've evolved historically, as well as some of the more recent trends in those industries, and kind of pull insights from them. So the two that are kind of most obvious and on very different ends of the cost spectrum are, on the one hand, the biopharma industry that has infrastructure for pretty large-scale culture of animal cells. And then on the other hand, go to something like the fermentation industry that we were talking about on one of the panels yesterday, where they're operating at hundreds of thousands of liters, and it's for food application. So sort of the stringency in terms of operational controls and all of that looks very different from a biopharma facility, but it's the same concept. In both cases, you're culturing cells of one type or another. And so the challenge and the goal really for this industry is to say, what can we take from each of these to find that middle ground and have something that supports cells of this type, but isn't so over-engineered that it's going to produce an exorbitant cost.
I'm just curious, for everybody who's in the audience, if you could put your hand up if you're working on the hardware side of scale-up of cells. Just a handful of people. So that's really interesting, because this represents a huge opportunity for the industry to partner, if those folks aren't here at this meeting, to think about bringing in the mammalian cell culture people who are working at the hardware side of things and look at what are the needs of the whole industry and what future technologies could they be developing, or applications or hardware. And that's one of the other topics that we wanted to touch on, was this idea of partnership between academia and industry. Amy, maybe you can give some examples. You said that you're on the board or the advisory board of Beyond. How has industry interaction worked for you and your lab at UCLA?
Well, in various ways, we definitely have interacted quite a bit with industry. My lab, a major focus of my lab is on technology development and in the mechanobiology space. So we've had some industry-sponsored research agreements and interaction with industry that hasn't progressed so far. There's been a lot of enthusiasm talking with research people on R&D teams, but then passing through the hierarchy to approve a collaboration with academia sometimes doesn't always pan out. So definitely we prioritize working on projects that have translatable potential. So we're very open to partnership with industry, but I think it can be challenging just with the different timescales of operation. And I know from the industry side as well, having a team of dozens of people focused on one question is quite different than academic labs, where typically there's one student who is learning how to be a scientist, who's working on the project. And so I think that those differences can be challenging. And then IP issues come up as well, which can be challenging to navigate as well. So those are some of the issues that we've encountered.
Didier, any thoughts?
Yeah, sure. As I mentioned earlier, Aleph has been started as a tech transfer from the university into the company. We acquired a license to transfer the know-how, technologies, patents developed by Professor Levenberg in the space of tissue engineering into food. And we do see at Aleph today two types of models for the collaboration with the academy, and we're working with quite a few universities. One model is the model we continue to implement with with the Technion, which is a research grant or a funded research agreement. And the other model is more a consulting. We're also working with 3-4 other labs in the world, in the US, in Europe, in Israel, for a consultancy, which is more being advisors or providing some inputs to the R&D we perform at Aleph Farms. I believe that the first model, which is the funded research model, should be structured well for it to work. At Aleph Farms, the way we look at it is that the research at the academy is really focused on exploratory research, on figuring out new ways to do things, kind of exploring new solutions and some specific ideas which are completely out of the box. And then when those ideas look like they work, we're kind of transferring them into Aleph Farms for actual development activities. And that's based on the fact that, as Amy mentioned, the way academy and university and the company work is very different. In the academy, you have very loose timelines. Budgets are sometimes not very well defined either. The team is evolving. The way the research is performed is not always very well documented and according to procedures which are in line with the industry standards. So it fits very well preliminary exploratory research. On the other hand, within the company, you're very much focused on achieving your goals, meeting your timeline budgets. So you're usually not really open to explore new things. Everything is within a framework which is contained by time, human resources, and money. So it's not an environment which is very supportive for just exploring new things and thinking out of the box, trying different approaches. So the complementary between both, I believe, is great, and the key is really to build the bridge right between the two, to make sure that the activities performed at the university on one hand can benefit for some flexibility and are not too much directed by the company, but on the other hand are relevant for the company. And then the company has the team on board for incorporating that research and transferring the research into actual development when it shows some promising results.
So we're here in Silicon Valley. The Semiconductor Research Consortium is a research consortium by the semiconductor companies, and they identify as an industry the groups of research that are problematic, that are pre-competitive, that cost a lot of money, take a lot of time. But if they could fix those things, the whole industry would jump forward by leaps and bounds. And they have a patent pool process. So the stuff goes into a patent pool, you pay an annual fee, you get to access that patent pool. Liz, is that something that the Good Food Institute has thought about? Do you think it's too early for that for this industry? Is it a model that could be productive for funding academic research in some of these areas?
Absolutely. I think there's a huge need for dedicated research centers that are hubs, knowledge hubs for industry and academia to work together, as well as folks from government labs and so forth. This is one of the best mechanisms that we have, and a model that's been pursued in many other industries for kind of addressing the so-called valley of death, this well-acknowledged concept in research that universities are really sort of the hotbed of innovation at what we call technology readiness levels 1 through 3. So fairly early stage technologies. Then once you have something kind of through prototype, demonstrated, ready to go to large-scale commercialization, it's easy to find investors who are there just to kind of take it to scale, right? It's been proven out. There's relatively little technical risk at that point. But in the middle of those two, there's kind of this chasm where it's not really clear, does this fall in the domain of something government should be funding through public funds as kind of a public good? Or is this something that's sort of within the tolerable risk profile of an investor? And so these industry, university, collaborative research centers really bridge that divide and make sure that there's kind of a direct handoff from some of this earlier stage research to real commercial application, that you've got a ready commercialization partner. And it can also reduce some of those transaction costs associated with an individual academic lab setting up a partnership with an individual company when there may be, internal shifting priorities and so forth. If you have established members of a research center or a consortium, then that, everyone's at the same table, and it reduces the kind of paperwork load and transactional costs.
I'm familiar with these manufacturing institutes. There are these grants out there for that. Have you looked into that for cell-based meats?
Yeah, absolutely. So the US government has developed a suite of these. There's more than a dozen now, advanced manufacturing institutes that are really intended to kind of be a sandbox for scale up and commercialization of new manufacturing. So there's one for biomaterials and biotechnology, quite a few different fields. And these are funded by a number of different US agencies, government agencies, depending on the institute. But that model is precisely something that has value here, and we're starting conversations not just with federal agencies that might be in a position to fund these, but also going to state governments, even local governments, and saying, look, this is an opportunity to really bring innovation to your jurisdiction, and be seen as a leader in this new pioneering field.
Great. I want to throw out the questions to the audience. So if you have a question, please raise your hand and a mic should come to you. While we're waiting for audience Q&A, Amy, what are the bottlenecks around scaffolds? You mentioned that you work on scaffolds, and I know there was an announcement by Innovators yesterday about their new company in mycelium scaffolding. What are some of the bottlenecks that you have to look at for scaffolding?
Well, I think as Didier mentioned, the fabrication of scaffolds in a scalable way that's compatible with a bioreactor, for example, is a major challenge. And so thinking about how you can pattern cells in a way that can be scaled up. 3D printing has achieved great successes in generating even like contracting ventricles of a heart. So functional organs. So it has a lot of promise, but scaling up how you deposit cells in a controlled spatial locations is still elusive. So I think that's one of the major challenges.
One of the others in terms of scale up, it would be the batch versus continuous fermentation. Is anybody looking at continuous fermentation for mammalian cell culture?
So in the literature, there are some research groups try to do continuous biomanufacture for like the human pluripotent stem cell or human other cells. But presently, I don't like that idea because, especially for biomedical applications, you produce one batch of cell, you need to do a lot of characterization, quality control. I think for the animal cells too. So for the cells keep changing, it's not like a chemical. You can do continuous biomanufacture and you do a mass spec, you know the structure. What about the cells? They keep changing. How you do continuous biomanufacture, the time affects the cell phenotype a lot. So I prefer the batch. Of course, the continuous biomanufacture can reduce the cost, obviously, but it's something you need to consider, the quality, quantity, and cost.
Didier, any thoughts on continuous culture?
Yeah, currently we do not use continuous culture. That's definitely an option which might become more relevant in the longer term. The questions are really, what are the issues you'd like to solve, and what are your bottlenecks in terms of cost and scale up? I believe that every company has. Each company has different technology, different platform, with different bottlenecks. we're talking about scaffolding versus non-scaffolding. I think that the type of cells is also very important. If you're using pluripotent stem cells or induced pluripotent stem cells, it's a completely different system you're building than if you're using, let's say, satellite cells or fibroblasts, which are partially differentiated. So I don't think that there is one good answer, one approach which is the right approach for scaling up. Each company really has to focus on solving its own bottlenecks according to the type of product and the technology it's implementing.
Great. So we have a few questions that have come up through Slido. So if we can bring these back up. Well, the first question was, where in the world do we think the first market for cell-based ag is going to become commercially available, and on what time frame? I've heard estimates for next year in Asia. Does anybody want to take a stab at that?
It's probably more commercial, so can we take it? Yeah, so we look at the three main geographical areas, the three main geographies as being the US Americas, Europe, and Asia Pacific. We believe that Europe might take longer time to clear cultured meat for marketing. In the US, there is some good progress thanks to GFI and the other companies doing great work here. And we've been active in the interactions with USDA, FDA. We plan on being even more active in the next couple of years. However, it might be that Asia, especially some countries which are very proactive, like Singapore, Hong Kong, Japan, might be the first to clear cultured meat for consumption.
Liz, do you agree?
I do, yeah. I think we've seen some countries really leading the way, showing their willingness to work with the industry to clear those regulatory hurdles, which I think is really promising and paves a path for other countries to follow suit. In terms of timing, you could go to market almost immediately with a product that is at small scale and high price point, right? Meat is not just one thing. There's not some magic number that we need to get to in terms of volume or price point to suddenly be competitive. There's a whole spectrum of products, and most of the cell-based meat companies have sort of a strategy that takes in mind, what that roadmap looks like going forward. Can we start in kind of a high-value, premium kind of rollout, and then as we, continue to iterate and refine our product, as well as get to increasing levels of scale, where does that take us in the years going forward?
So am I right that there is no commercially available product from anybody
Correct.
right now? Now, if I was taking the Wall Street Journal on a tour of cell-based meat labs, which companies could I go to and actually taste the sample right now?
Quite a few of them. Yeah, so a lot of these companies have done several rounds of both. They do internal kind of taste testing of their products and are working on sort of the food science product development side, not just the cell biology, but also doing tastings for folks like investors and so forth. So quite a few companies have done tastings and prototypes, and some of them have been public, open to food journalists and so forth.
So I'm more familiar with the ones here in the Bay Area. I know Memphis Meats is at that stage. Finless Foods is at that stage.
Wild Type recently did a salmon, cell-based salmon 6 ways tasting dinner.
Where's Wild Type based?
Yeah, also here.
Oh, great. I haven't heard of Wild Type before. Any others that folks know? Have you done demos for journalists?
Yeah, sure.
Investors?
I think it has been published recently on CNN, CBS, last couple of weeks. But we had a few testing events in the past. In the quick movie I presented at the beginning, we had some samples of our meat, which are the initial prototypes we had released at the end of last year. So yes, definitely.
Great.
But it's not yet approved for marketing. So anyone testing it has to sign a waiver, and all the things, which is the same probably for all the companies. It's not yet available.
Well, maybe it just could become a standard for the industry, like the software licenses that you click through. We'll just start doing that for food soon. It'll be fine. Any other questions from the audience? Maybe if we can bring up Slido again and we can see if we've got any other questions on Slido. So thinking back about policy, what policies do you think we could put in place that would encourage more funding of research in this area, would encourage more infrastructure to be put in place for this area, or would encourage more interaction between industry and academia? Any ideas on creative policy ideas? Amy?
I can't really speak to policy, but funding organizations like the National Science Foundation, for example, I think is a prime target to support this kind of research. One of their mandates, is to promote industry academia collaborations, and there's certain calls for their funding where they really try to emphasize that aspect of a proposal or project as well. I think that's really important. They're also one of their missions is to train the next generation of future researchers and scientists as well, which I think is also really critical for the industry as well. So at the moment, there's no specific call for proposals on cell-based meat. There are some funding mechanisms that are general enough that those projects could fall under those other calls for proposal. But I think having something specific to the industry, to the field, funded by an agency like NSF would be very advantageous.
Amy, I'm inspired by so many young entrepreneurs starting cell-based meat companies and starting food companies and starting synthetic biology companies. How are you seeing the inflow of students and postdocs wanting to come to your lab because that's what you're working on?
It's intense. Yeah, it's great. I've been surprised. I mean, we got into this thanks to the GFI. We were awarded one of the inaugural research grants earlier this year, and that brought more visibility to our group, and it's been surprising already the number of students who've been very interested in the project. In fact, the whole reason my lab got into this project was because of a student, Stephanie Kavesky, who's here somewhere as well, who was very passionate about the field and was able to see my background in mechanobiology, science, and food, and kind of pitch this project. And so I think that demonstrates that, those students who are passionate about this kind of research, that there's ripe opportunities to find other professors, research groups working on related topics, and help to persuade them to embark on that kind of research. So even here at the conference, I've met many interested students. So yeah, I encourage any others to reach out to us.
Leo, you're more known for your work in human cell culture, maybe less known for your work in this area, but are you getting an influx of students wanting to work for you and in your lab?
The human cell and the animal cell, there's really not too much difference. So students mainly work on the human cells, but we can almost directly translate into the animals. So we have a lot of students work on human cells, but not animal cells.
Great. Liz, any thoughts?
Yeah, I just wanted to add, I think historically one of the big challenges for getting funding for this topic area is that it doesn't fall neatly within any of our existing disciplines. And so, the competitive grant program that GFI's been doing for now onto our second year that Bruce mentioned this morning is really intended just to be sort of very early stage catalytic, allow labs that have sort of the fundamental tools at their disposal to address some of these challenges, but haven't yet explored this application area, to develop some preliminary data, show some tractability with their research, and then be able to have sort of a more complete, compelling picture to bring to some of these larger research funding agencies and have success there.
Great. I think we have time for one more question, so we can just bring the questions back up on the screen. This question was about hardware in particular, and lots of discussion about bioreactor innovation to reduce costs. how are reducing costs of expensive media like growth factors? So how do we get the costs of the media? Didier, any thoughts there?
Yeah, I think that it's a good question, but it's not necessarily the right question. I think that the question is how to reduce the cost of the production process as a whole, and the medium is one aspect of it, just one parameter. You can recycle the medium and reuse part of the growth factors which haven't been used to reduce the total cost. You can replace growth factors by other signals as part of the production process. So to a question, there are many different approaches. I believe that part of the growth factors will need to be produced recombinantly, meaning with relatively expensive production methods. And here the question is related to what Liz said at the beginning regarding going more for biopharma partners or fermenters, large fermenters. So we're working on a combined approach, and we believe that only a combined approach, systematic approach, can help the companies and the industry reach its goals in terms of costs.
Great, thank you.
I'll just give one plug. Anyone who wants to go deep into cell culture media costs, you can find on our website a white paper doing a deep dive cost analysis with some various scenarios for scale up of cell culture media.
Wonderful. Liz, Leo, Amy, and Didier, let's thank all of our panelists, and thank you as well.
Thank you.